A significant percentage of biopharmaceuticals are protein-based therapeutics. In the past 5 years or so, monoclonal antibodies and fusion proteins made up 50% of FDA-approved biopharmaceuticals. Manufacturing of these protein drugs is actually a very complex process, making it almost unavoidable to introduce some sort of diversity in the end products that is undesirable but realistically difficult to prevent, simply because of the inherent biomanufacturing process. For example, upstream processing involves culturing of cells in bioreactors that are at set controlled conditions. So any changes in that physiochemical environment, such as pH, temperature, dissolved oxygen, and such, can negatively affect the quality attributes of the product. Downstream processing, on the other hand, can also be a source for heterogeneity in the product. At this stage of biomanufacturing, you have the harvesting of cell cultures, and then you're performing several complex purification and filtration steps to try to remove different types of impurities. So sometimes harsh conditions can be used during this and can additionally expose the protein to various types of modifications, again, negatively impacting your protein product. The formation of protein aggregates during monoclonal antibody production is a very good example of an issue that is influenced by the bioproduction process. This results in not only product loss, but also affects product safety. So just from the inherent complexity of manufacturing biologics, we really need analytical methods that can thoroughly characterize a drug product to help mitigate some of these issues that arise during bioprocessing. So characterization is important not only from the primary structure level, amino acid sequences, but even to higher order structure and conformation. Popular or mainstream methods a lot of times can be broadly categorized under chromatography, spectrophotometric, or electrophoretic techniques. But in the most recent years, the technological evolution of mass spectrometry has made it a significant asset in biotechnology. MS-based workflows can be used to characterize intact proteins with very minimal sample prep, primary sequences, post-translational modifications such as glycosylation, oxidation, deamidation, isomerization, disulfide leakages, and higher order structures. Additionally, product and process-related impurities can also be identified and characterized. A significant advantage of MS-based workflows is that several attributes can simultaneously be monitored in a single data set. In this series of short videos, we'll illustrate the analytical usefulness of MS in quality control of protein therapeutics, especially when assessing product identification, impurities, so both product-related and process-related, and product structure and variants. Additionally, we'll cover raw materials testing and culture media analysis using MS approaches. Each module is designed to be topic-specific and focuses on one particular attribute class and the advantage of utilizing mass spectrometry as the analytical tool. Lessons within the module will include description on how to execute each assay, translating your traditional methods to an MS-based one, and the core MS fundamentals associated with the assay.